Thank you for that kind introduction. I love movies too, by the way.
Oh, thank you.
I hope to make this informal. I came here hopefully to actually explain some things about what we do in a simple, straightforward way. And I'm happy if I seem unclear at some point, it's okay. Step up, put up your hand as we're going. If it's a short question, no problem at all. What I've tried to do in organizing this talk is to give you three basic updates. One is about a topic that I spent a fair number of years on pertaining to the organization of the visual pathways, in particular the visual field maps in the human brain. It's now been about 15 years that we've been able to measure those, and there's been a lot of progress in understanding the organization of the visual field, which I will explain what a visual field map is and how we measure it as we go along. There's been a lot of progress in that field, and I thought I'd give you an update on that and where things stand. And then the next thing I'm going to do, that, by the way, I believe, listening to the other speakers, may bear on some issues concerning the computational architectures that one might consider for visual recognition. The second thing, I'm a person who's quite attracted to applications. And I've become quite interested in the question of to what extent the human brain reorganizes, the adult human brain might reorganize. And I just wanted to give you a sense of where that field stands also. And I'm glad Dr. Schutz is here. She and I are actually co-authors on one controversial paper in this area. And I wanted to give you a little sense of, I think there's a certain zeitgeist, a certain feeling that one has about the extent of plasticity in the adult human brain. And I just wanted to challenge that a little bit in the middle part of the talk. And then in the third part of the talk, I'm going to talk about something completely, again, development and plasticity, but a different imaging modality, this time pertaining to measurements of the white matter in the human brain and the white matter tracts using a technology that's called diffusion tensor imaging, another MR modality. And so the tough part of this talk is that there's a lot of stuff that'll fly past you. The good news is if you don't like what I'm talking about at one moment, just wait 10 minutes. It'll be something completely different. For those of you who are lucky enough not to have been in an MR scanner, this is what one looks like. And usually as a subject, we'll take a graduate student, or the graduate students will take me, and say, go lay down here, put your head over here, and then this table rolls into the middle of the magnet. And for visual stimuli, when you're not just measuring function, but when you're measuring activity in the brain in a functional scan, we'll look at a display on a screen like this. It'll be turned around so that you can see it. And this is an example of a subject. We build all kinds of little pieces of gear. This is a Ferris-free binocular rig with a mirror over here. So this fellow's looking up through these binoculars, and there's a mirror here, so his gaze is down towards his feet, and that brings him optically very close to the screen so he can get a good high-resolution view. As you know, the occipital lobes at the back of your head are where your visual areas are, and so there are various little surface coils that can focus in on one part of the brain or another if you want to emphasize a high-resolution signal. And just so you know, people often ask, what are the resolutions that we can measure activity in the human brain with MR? The spatial resolution. And the conventional one, if you just open up the literature and look, is on the order of 3 millimeters on a side. That's completely standard at this point. We've published, and other groups are publishing at 1 millimeter on a side. So, you start to take advantage and notice when there's an order of magnitude, or in that case, 27-fold increase in spatial resolution. And that will be completely common in the next 5 years, I'd say. People do more invasive things, and with some futuristic ideas, I think we'll be down to half millimeter, 500 microns, surely within the next 10 years, routinely. And it'll still be slow, but it'll be, because it's the blood, but it'll be highly spatially localized. Now, again, most of you probably don't do this sort of thing for a living. I just want to give you a sense of the kinds of details that we pay attention to in analyzing the data. And when we look at activity in the brain, and it might be a little guide for you when you see different kinds of results. Not all brain data, neuroimaging data, have to be analyzed in the same way. It's okay to do experiments for different purposes in different fields. Sometimes you're just wondering for some phenomena, is it the frontal lobes or the parietal lobes? Okay, you don't have to do everything in detail. But if you're interested to get down to the level of neurons and pieces of gray matter that might interest a physiologist like Ed, say, well, we got to worry a little bit more. So here's the story on that. This is just a conventional, MR slice, an axial slice of a brain. And when it comes out of the scanner, if you're a MR physicist, you get a set of sample points like this, and you might treat it as an image file. And normally, when you think of an image file, you think of neighborhood relationships. As I'm a voxel here, and I look to the right, look to the left, look up, look down, and that's who are my adjacent voxels. And that's a kind of a natural move. But if you're a neuroscientist and you look at a pair of voxels like this one and this one, the two red ones, well, they're next to one another in the image file, but they're not next to one another in the brain. The brain, the gray matter that you see curving around here, forms a surface that'll often dive quite deep into a sulcus like this. And so although these 2 points are next to one another in the image file, they're not next to one another in the brain. So you want to make sure that if you average or smooth or look at data in neighborhood relationships, you don't use the matrix that the typical physicist would do. But in fact, you want to segment out the brain and find the boundary between the gray matter and the white matter, as shown here by the green curve. And in that case, your connectivity relationship, again shown by the blue as it is over there, the connectivity relationship puts this voxel here on the other side of the sulcus with this one, from this one here. So you would never average across that unless you really didn't care about the spatial resolution that you had obtained. So one of the things we do, and a number of groups do, and you could look for this in different talks in different ways, is to carefully segment out the white matter and the gray matter in order to preserve the topology, the spatial relationships between points that we measure. That then leads naturally, once you do that a lot, white matter and gray matter segmented out. By the way, I'm telling you about this partly because I want you, I want you to know that we do this, but also because I want you to know that the process of doing this is no good. It takes us a long time. And I don't like that because I don't want to do this a long time. I don't like to ask graduate students, go spend a week segmenting a brain. That's just annoying. And you'd think at this point in time with all the papers on segmentation that we'd have wonderful tools for doing this. Not so good yet. And so those of you who think, what is this? Some of you are sitting out there thinking, what is this guy talking about? I could do that just great. Meet me afterwards. I'm ready. I'm ready to try it. And I'm honest. I'm not looking to say you can't do it. If you can, I am really happy. I'm not trying to say negative things. I'm always looking for a good tool on that. So far, not so good. Okay, so now once you have segmentations like this, it becomes then straightforward. The part of the field that's quite good to go is once you have this set of surfaces identified, you can build 3D visualizations of these surfaces very easily using well-understood techniques from computer science. And particularly, I want to say the VTK toolkit has been very useful to us. And you can do standard visualization operations. It's hard to see down in these sulci and so forth. So we often just take that surface once it's segmented like this and just sort of blow some air into it and puff it up. And that makes it much easier to visualize the surface and see it. And then we'll code the parts that are sulci, that are indented, as dark, and the parts that are gyri as light, just so that we know roughly where we are on these things. And I'm showing you this because I'm going to be showing you pictures with data like that on it through the talk. So I wanted you to give a sense that those weren't kind of average blurred ones. They were very carefully segmented ones that we just make it easy to visualize with. Okay, now, let me introduce those of you who don't know about this to visual field maps. And the most famous visual field map is the one in primary visual cortex, or area V1. And here it's shown, actually, let me get you started on another one just a little bit. See, this is one of these brains over here, coded just the way we are. In primary visual cortex, this is a medial view, right? You can see this is one half of the brain, the right hemisphere in this case, medial view. And this sulcus over here is the calcarine sulcus, and that's the one where primary visual cortex is. Now, this is a little blow-up. This is from Jonathan Horton This is a posterior part, the occipital lobe of a human brain, and he's drawn this in such a way that the calcarine sulcus is sort of exposed so that you can see it fairly easily. And the visual field map in calcarine sulcus is very well known, as discovered from head injuries over the last 100 years. First off, the left, you can see that we're looking at the middle of the brain here and the left hemisphere. And the left hemisphere is stimulated only by visual signals that are in the right visual field. So this part over here doesn't matter to this map. In the left hemisphere, it's the right visual field. The horizontal meridian drives stimulation at the bottom of the sulcus, the lower vertical meridian at the top lip, the upper vertical meridian at the lower lip, and then the foveal projection, the central 2 or 3 degrees of your visual field, has a very large expanded representation at the back over there. And that's because you have many, many more cone photoreceptors in the central part of your visual field, very high density, and when they arrive at cortex, they finally expand on out and take the cortical area that reflects the high density of cones in the center of your visual field. So about, I guess it's almost 15 years ago now, Steve Engel and I and some others started using MR to map these visual fields. And the first thing that was really Steve's idea was to use a very simple stimulus, a kind of a polar representation of the world that's quite natural to sort of look at the activities in the human brain. And one kind of map you therefore might measure is to have somebody fixate here at this red dot and then have this ring expand in this way. And if you maintain your fixation here while the ring is expanding, see, I know what's happening in your brains. It's all pretty much the same at this part. When it starts out in the center, the activity is here, and then it travels along the calcarine sulcus, mapping the eccentricity in this one of the dozens and dozens of brains that we've looked at. Now, in the actual MR, this is speeded up. In the actual MR, it's a much slower process because it goes through the vasculature. But just to make it visually obvious, we run this thing at this speed. So that gives you the eccentricity portion of the map. And of course, it's quite possible. And we represent this sometimes just as the same data. We represent them as a color map where we would code the fovea as red, green, and then as we go peripherally, green, cyan, blue, red, so forth like this. And you can see the eccentricity map on the brain in that way. Well, in the early days, I think we first published on this in maybe 94. So that was really exciting, right? And so you felt good about it back in those days. These days, it's so routine that we only get annoyed when it fails to work. And I've always thought that was unfair. There's something wrong about taking away the joy of seeing that again and again. But anyway, that's how it goes. You can also take a wedge and rotate it around and get the other dimension of the map. So here you can see blue to green to red. Just as I told you, the upper vertical meridian is over here, the horizontal meridian in the bottom of the sulcus, and the red over here at the upper lip of the sulcus. And you can see, therefore, the map in primary visual cortex just from those two kinds of measurements. The other thing you can see, if you just look at the colors here for a moment, that it goes from blue to green, back to over here, and then back to blue again. And in fact, there's another quarter visual field represented here, and another regular map over here. And in fact, there's a series of these maps. And we know now the field, by we, I don't mean me personally or just my group. We've discovered a few of them, but there's good agreement on about 11 of these maps. And let me just give you a sense of them and a little bit or layout on the human brain of where they are. Their location differs slightly, it differs significantly from the macaque brain where most of the work was originally done. I've already shown you primary visual cortex is this dark blue. This little bit surrounding it over here, there's a quarter field, and then down at the bottom side, there's another quarter field over there. That's V2, the second visual area that was been represented in various slides here today. Here's the third visual map that surrounds it once again over there. The ventral stream that people, are very fond of for describing for image processing runs down this way. In human, it appears, we have argued, and this is somewhat controversial, but it seems more people are signing on to us than not, that there's a fourth visual map that in the human is completely confined to the ventral surface. In macaque, it surrounds V1 as V2 and V3 do. But in human, the fourth map that appears to be essential for color vision, in the sense that if that map is destroyed, you become achromatopsic, which neurologists have shown. Samir Zeki has been on this for many years and so forth. And we were able to confirm that in our own way over the last several years. And then there's another set of maps down here. Regions over here, if they are damaged, you lose your ability to, for example, recognize faces, a condition known as prosopagnosia. In my own work, we've been looking at alexia, which is the loss of the ability to read. Again, can be caused by damage either down here, where there will be other maps, we just haven't found them yet, or in fact damage in the callosum, which I'll tell you about later. This region over here is motion-selective cortex. In human, we can't segment out MT from MST repeatedly, though there have been a couple of reports of the ability to segment out these areas, including from our group. And then over here in the intraparietal sulcus, there's a whole set of areas. These are a few of them. Marty Serino, Dave Heeger, others are reporting areas up there all the time. And they are particularly responsive as you change your attention from one task to another and task switching and so forth. All of these areas here, these were the conservative ones. These will all be filled in with visual field maps as we get better and better at measuring them, and there are a number of ways that we've learned to measure better, even in the last 5 years. And so we're getting a better picture of both the maps and kind of where they stand. Now, and something about what they do. Sorry. Now, one of the questions you might ask that we've asked, and that I want to really credit Rafi Malik at the Weitzman Institute for first asking, is there some organization to the maps themselves? Is there something about them? Is it just like one map to the next around here? Or do the maps kind of come together in groups? And I actually really admire Rafi and Uri Hasson and their group for asking the question. We don't like their answer, but they did it, I think, raised it and made an interesting proposal, and we followed them in that domain. It's too hard to clip on. I'm just shoving it in my pocket. Okay, and let me tell you what our answer to that is, and we continue to work on this, and this is, instead of everybody agreeing, this is one of those things that we have a view on, and that's our opinion, and that's the truth. So I've already told you about these expanding ring maps that go up calcarein. When you measure these maps a lot, one of the things that you'll see, and again, nobody disagrees on this point here, is that out here in the ventral surface, where I was telling you about face representations and letters and so forth, you can see, I mean, look, you don't have to be a rocket scientist. You see this little red spot, and then there's green, and then there's blue, and then out here it goes on to magenta. It just looks like another little band of eccentricity. And this is centimeters, about 4 centimeters away from the foveal representation at V1. So this little foveal representation over here is plainly not the same one as the V1 representation, and there's a nice little eccentricity map right around it. And in fact, that suggested to us that this notion of an eccentricity map and then several other maps connected to it could be a general principle. In fact, there are 2 complete visual field maps over here. I'm not even going to talk about this other little foveal representation out here. That was beyond the plane, right at the edge of the plane of our measurements. And what we see when we sort of summarize the data from many, many different experiments, and I'll show you this as real data in a moment, is that around V1, V2, V3, in the 4th visual area, there's a common eccentricity map. And then it subdivides into different, by angle, into different field maps. The other one we're just looking at on the ventral surface down here is another little cluster of maps. In the intraparietal sulcus, there are actually 2 clusters we've been able to identify. Around MT, there's yet another cluster. Around this lateral occipital region, there will be another cluster. So we think that the basic organization of these maps is something that's real also. It's not one map to the next, and when genetics or evolution kicks in, you fire off one new map and off you go, but that the computational structure probably has to do with these little groups of maps that share a common eccentricity field and then subdivide by their angular orientation. If you take cortex and you just flatten it out, you can see this wasn't the kind of theory that this isn't really complex theory. We're pretty good at just flattening brains computationally also, and you can just see this coming out this way. Here's the ventral maps. Here's the map up here in intraparietal and so forth. Here's the MT map. You can see this thing. It's not so perfect that it's just like the cartoon, but you can see it in the flattened image itself. And so we're quite interested in this, and we think this may have implications for connectivity and a variety of things. And the locations of these maps just on the 3D brain, where I prefer to see them, is just shown here by the sequence. Okay, so in summary, that was my update on maps. Visual cortex can be subdivided into a multiplicity of visual field maps. We're good on about 12 of them, 11, 12 right now. The field's not done. I might be done with that in my life, but the field's going to keep reporting these. About every 6 months there's a new map reported from somewhere. Beyond V1 and V2, and maybe MT, and maybe V3A, the homology between macaque and human maps is quite uncertain, quite uncertain. And in fact, I don't think this is arrogant to say that right now it's easier to measure visual field maps in human than it is in macaque for a variety of reasons. Mostly just the process of going and poking in the electrode and getting the cells excited in an anesthetized animal beyond V1 and V2 is just really hard, whereas you take an undergraduate and shove them in there and they come out for a cookie and they're happy. So they go and they tell their parents that they're getting a good education. It's good stuff. So it just seems that we've actually, in that part of the field, for that kind of measurement, to be easier to do on human. And so I think that's a good thing. It's an advance, and hope that we'll learn more over the next couple of years. Now, the next bit I wanted to talk to you about is plasticity. And let me just start out by saying, please, nobody leave here with saying, oh, that knucklehead Wandell, he doesn't think there's any plasticity in the brain. I mean, that's just, please don't say that. I've given this talk a couple of times, and people often start their questions at the end by saying, so why don't you think there's plasticity in there? Please, I think there's a lot of plasticity. And the question is, where are things plastic? When are they plastic? What are the mechanisms of plasticity and so forth? And I think there's a good fight looming about the extent and nature and conditions in which one observes plasticity. And that's what I wanted to talk about. And I'm going to tell you 2 stories. The first is about this fellow here, Mike May, who Ione Fine, who's now at UW, when she was at San Diego, she brought Mike to our lab. I continued to collaborate with Mike on a variety of things. Mike is a remarkable fellow. He, as a child, at 3 years of age, he had a chemical accident, a little explosion in the garage of his folks' house. And one eye was just completely blown out. And the other eye, the cornea and lens, were very seriously damaged. And most importantly, the limbic stem cells that are needed to replenish the cells that keep the cornea and lens clear were destroyed. So that although his retina was intact in that eye, no cornea and lens transplant could be done so that he could have good contrast vision. And therefore, he lived essentially as a blind man from the age of 3 to 46, at which point in time it was possible to do stem cell replacement and to put in a good cornea and lens, which he did. It was done here in an operation in San Francisco. And so here we had a fellow who had lived for 43 years, from the age of a child to the age of 46, without any contrast vision to speak of into his visual pathways through this eye. And then after this operation, he had it. Now, most cases, there's been in the history, in the written record, probably about 30 cases of kind of miracle cures of this sort where sight is restored after long periods of effective blindness. And most of them, you should know, turn out very badly. People end up depressed. They never get really good sight back. And so I was a little worried about Mike, but Mike is a very special character. You should know amongst, in many ways, especially that. But one of the things, just to put a punctuation mark, an exclamation mark on it, Mike holds the downhill speed skiing record for a blind guy, 60 miles an hour. He won the Olympics, the Special Olympics in this category. And if you wonder how a blind guy goes down 60 miles an hour down a ski slope, let me tell you, he has the world gold medal Olympic skier in front of him saying, go right, go left, jump, stuff like that. Just really, so this is not, a shy, awkward guy who's a little timid about stuff. And so Ione brought him up to us at Stanford so that we could help, because although there had been a number of cases like this reported, Oliver Sacks has a case described in the New Yorker like this, no one had actually seen the brain of such an individual, and we actually just made another round of measurements on him just a few months ago. We've been tracking him over the years. And let me say, it's a pretty clear case that if this is the normal layout for the eccentricity map, which, as I tell you, is quite easy to measure in normals again and again, in Mike's brain, it has a very different layout. Look at the large representation of the periphery compared to the normal, and look at the odd placement of the fovea, which is way over here instead of all the way back at the occipital pole. It actually wraps around the occipital pole in most people. It's very unusual. Now, this damage happened when he was 3. So he would have grown up, like all the rest of you, to look something like this, but with those 43 years of experience, starting at a young age, he grew up very differently. Clear case of plasticity. We published on other cases of plasticity. Nothing. This is developmental plasticity. Nothing surprising about that. You should know that Mike, even though he has a good image on his retina, and we can get an ERG out of his retina, when you ask him, how do things look? What do you see? He says, well, I'm getting smarter at it. And what he means by that is when I look at something like this, or you look at me as like this, and I walk around, or as you have Asimo looking at these things, right? Asimo learns about these things and comes to some efficient conclusion. Mike still has to reason. He sees something from one point of view, and he walks around here and says, I can see the color didn't change, the shading changed, but he's faster and faster at reasoning. But he still is reasoning. It's not automated for him. He doesn't recognize facial expressions very well. All the things that we take for granted that we do in normal visual object recognition, he doesn't do well. Okay, so,
does he have acuity?
Does he have acuity? His acuity, you would see out to about 30 cycles per degree. He's good out to about 6 cycles per degree. But it's not because of the image. So in other words, we can see the image in the back of his eye, but he can't see it. And he's apparently just lost parts of his visual pathway that are not reconstituted. In fact, we can't get a decent response out of his ventral occipital lobe. We get an excellent response out of motion selective cortex in his brain. Okay, so lots of stuff. Sorry, is that me?
When you talk about the other 30 patients, some of whom were depressed once they got their vision, was it because they were so confused or so having such difficulty learning this process? Was that part of their...
People speculate about that. I think it's important, particularly, as medicine advances, to be alert to this sort of thing. And there are 2 sentences that I was particularly struck by in reading that literature. One is a fellow said, I had lived my entire life as a very successful blind person, and now I'm not such a good sighted person, and I just find that hard to deal with. And the other thing is our expectations of people, what they can do and what they'll notice and so forth, is based on, if you see that they're sighted, you say, well, why didn't you say hi to me? I have a close colleague, Jack Nachmias, who's had vision his whole life, and he would walk, but very low vision. And he'd walk around the halls, and he wouldn't say hi to a graduate student or something, and they say, "Oh, well, he's a mean guy." Right? And I think the social context and the actual, expectations and how you feel about yourself change. And I should say, there was an Italian doctor who did a lot of these miracle cures, and 2 out of the 15 patients that he did, that he returned contrast vision to, suicided. So it was very serious. It's just, and I think if they'd been more alert, now we know, so it shouldn't happen again. Okay. At Dr. Schutz's lab back in Tübingen with Nikos Logothetis and others, this issue of plasticity seemed like a good way to finish out my career. I just thought, boy, I like this plasticity stuff, and it could matter, and it could matter in the clinic, and there's a lot of places it could matter. And what I'd really like to be able to do, and what Nikos Logothetis really wanted to do, thought, if we could get the pharmacology of this plasticity under control, we might be able to have an impact on people who, for example, lost part of their vision from stroke, or for whatever reason, or had retinal dysfunction and lost part of their vision. And we decided to use the Macaque model. This is, by the way, to scale. Human brain, Macaque brain. If you think that there's no difference across those scale, I don't know, whatever. I think there must be a difference. Let's put it that way. So here's Nikos's first rig. It was really a cool 4.7 Tesla magnet. He got the bore with great effort vertically aligned like this. He could put the monkeys in there and run them anesthetized. So we could do these visual field mapping experiments on them because their eyes weren't darting around. We didn't have to keep their eyes still. And with Elisa Brewer at Nikos's shop, we could get these same visual field maps out in the macaque. Here's the foveal representation near periphery, and then further in the periphery over here. Running across this part of the brain in the macaque is called the operculum. And they're perfectly fine signals, and Nikos built a remarkable rig. And then Stelios Smirnakis, who was a postdoc with Nikos and Elisa and the rest of us, thought, okay, look, let's now lesion the retina bilaterally. So here, left and right eyes, lesion, and he did that with a laser shot. Photocoagulation it's called, but it's the laser, lesion in the corresponding place. And that will block stimulation from the retina that would normally get to this piece of visual cortex in this map over here. And sure enough, he did that, and in fact, we couldn't see any significant signals coming out of there. It was a blind spot. We call this the lesion projection zone, because that's where the lesion projects to in cortex. And we thought, well, let's wait, because if you pick up a Psych 1 text, or you pick up your newspaper, or wherever you go, the general thinking is, there's a lot of filling in cortex, and you go and you make a measurement like this, and the signals are just going to start to fill in some magic way. And nobody had actually shown that it was going to help performance. We thought, but we could do that. Okay, I know how to do that, and so forth. But we thought we'd get this thing, and we'd get it under control. And because it was in an animal model, we could slow it with some drugs, or we could speed it up with other drugs. We might find training regimes and so forth. And that was our goal. And the darndest thing happened. We did this measurement. We did it on 4 different animals, and they all looked like this one. That is, we measured over time, and this is many months that we waited. That there was just really no significant change that we could pick up at this resolution, and the resolution we think was good to 1 millimeter in the filling in of that region of V1. We just couldn't see it. And we spent so much time and so many man-years on this project and worked on it so hard. And it went against what was largely the story in the literature, that we, instead of just saying, well, we couldn't replicate and burying it, we went and we actually published it. And I should say, maybe I should say one more thing. I should have said one more thing. I'm sorry. You might be thinking the same thing that we thought after we first saw those bold results, which is that, what this really proves is that bold is a lousy method for measuring activity in the brain. And that would have been fair. We did acknowledge that might have been possible. And Nikos is, I didn't do this, Nikos is capable of doing basically anything. And he put a linear array, single unit measurements in there, and actually measured across in the lesion projection zone and into healthy cortex. And the single units in healthy cortex, he could get a perfectly fine increase in spiking activity when he put on a big, wildly flickering field like this. But in this region over here, where the lesion projection zone, It was basically flatlined except for these occasional little spikes at onset and offset. Those are there all the time, whether you lesion, whether you don't lesion. They're just projections in from the lateral side. But there's no sustained activity through the lesion projection zone measured by single units either. And in fact, when you measure across the linear array and say, where does a spiking stop and where does a bold signal stop, they agree quite well. And so we ended up publishing that result. And the thing I wanted to do is to tell you that, look, plasticity is perfectly fine. I'm sure there's synaptic plasticity. I have no doubts about this. I have no doubts of plasticity on the orders of 10 microns, 50 microns, 100 microns. What do I know? Okay? But the notion that visual cortex could reorganize to fill in over a centimeter of empty cortex in the space of a couple of months by some kind of rewiring did not pan out in our experiments. And that runs counter to some of the things that you'll read from other groups and other places and times. And I just wanted to alert you that there's a fight either happening or about to happen, or I don't know how much of an impact we've had here. But we think that there's some serious issues there. And I particularly mention it because there are companies now, a wide array of neuroscience and neurology companies who will sell to you a little training box, $5,000, and you can, if you're blind, you do this training and you will become sighted. And we know that this is going to work because we know that there's plasticity in the adult human cortex, and just train this, and if it doesn't work, it's probably because you didn't work hard enough. And I see that kind of stuff, and it annoys me. And so I'm telling you about it because I'm annoyed. And now I'm feeling better, and so I'm going to move on. Okay, last bit of the talk pertains to another imaging technology, another method that we've been working on hard. It's based on a classic, and the goal of the method is to learn more about the white matter, the connective tissue that forms the core of the brain, of the human brain. And particularly, we're focusing on the white matter connections that are cortical. And I'll tell you a little bit about that. And our goal is to be able to identify the pathways in the brain and to understand what signals they're carrying, to measure their health and integrity, to understand the relationship between the health of these pathways and various performance deficits, and basically fill in a lot of our knowledge about how signals are carried between cortical regions in the brain. Now, I should say that when I say the white matter, for somebody like Ed Callaway, he's normally working at a resolution that's much finer than we can obtain. Or when you think of white matter axons, it's much finer than the resolution that we would measure with an MR signal like this. But you should know that the big white matter bundles that travel from one piece of cortex to another over a large distance, they don't travel, every axon gets its own way. They travel in big bundles. And there's connective tissue, there's infrastructure, there's a whole bunch of stuff that goes together. And they don't go just every axon has to figure out where it goes and, Godspeed. It's not much like that. And I should say that Dr. Schutz and Breitenberg do a very nice job in many of their published papers describing a kind of hierarchical structure. And I'm so sorry I wasn't here yesterday, but I'm guessing you again told the story of the very short pathways and the U fibers and the longer pathways and counting them and analyzing them and so forth. And that's very important work. It's been very important to us. And we think about that work from the point of view of, well, what can we measure? And we're measuring the really big stuff. We're not measuring the vast majority of connections that are very short. We're measuring the big ones. And these are the large fascicles that'll be 500 microns to a couple of millimeters across, carrying tens or hundreds of thousands of fibers at fairly large distances. And so when I say fiber or estimate of a fiber, that's what I'm talking about. And I'm usually calling them fascicles or fiber groups. Okay. So what's the principle? Well, the measurement method in MR to estimate these is very simple and very old, and it's based on the following idea. Here's a bit of optic nerve, sort of classic, all the optic nerve fibers headed up towards the brain here. And you can imagine that if you're a water molecule and you're diffusing around, it's not that hard to diffuse to go with the flow. They're all headed one way. You might diffuse fairly far along the direction of the optic nerve. On the other hand, if you're trying to go across the membranes like this, you're a water molecule, and you try to step through a membrane and say, I'm a membrane, I'm a water molecule. You stop, I stay here. That's just, water doesn't go through the membrane that easily. So if you could measure how far water diffuses in different directions, you'd get a sense that it would diffuse far this way, not far this way, and you could use that set of directional measurements to make an inference about how the fiber path is traveling. Okay, so diffusion-weighted imaging has been around for 30 years, and it gives you a measure of how far water diffuses in any particular direction that you set up your gradients. And classically, in medical practice, people would set up their gradients in 3 directions. And then starting in the late 90s or mid-90s, people realized, if we measured in 6 directions, we could actually kind of fill in the whole surface, maybe approximate it by an ellipsoid. Hence, diffusion tensor imaging was born. If we measured in more directions, we could get finer measurements of the shapes of these diffusion surfaces and learn even more. And that's the basic story of this field. Now, just to compare the diffusion tensor imaging data with conventional images, at each point in a conventional image, you get an intensity, how light or how dark it is. In the diffusion data, at each point, you get a surface. And so this is the kind of thing I always would have imagined would have intrigued Roger Shepard, who hired me at Stanford and my colleague for many years. It's got a beautiful geometry to it. It's first got the local geometry of the diffusion surface itself, and then the geometry of how these shapes and surfaces together might combine. And in the last few minutes, I'll just take you through some of the surprising results and abilities to make these measurements. Now, I should say, neuroscientists are very practical people. They kind of want to get going on stuff. And it wasn't really until the mid-90s that this kind of measurement became clear that you could do it and that you could get something out of it. But the math, the statistics of these shapes and so forth, even now isn't fully worked out. But nonetheless, a bunch of neuroscientists went forward and said, you know what, I know I've got these shapes. Let me do something simple. Let me just summarize the shape by one parameter. I know I've got a bunch of them, but it's too many. I just can't handle it. But I could deal with one. So let me summarize the shape, and here's one way to summarize it. This is called the fractional anisotropy of the shape, in which we say, look, how spherical is this, or how distended, how eccentric is it? And I'm going to color code this piece of the brain based, here's a standard T1, and here's the same part of the brain, but now I'm going to measure it by the diffusion tensor and how spherical or extended it is. And you can see that this picture, same piece of brain, looks really different from this picture. And that's the kind of thing biologists love and say, well, is there more structure in here that I might learn about the brain from the light and dark patterns here than here? And sure enough, it's like the strangest damn result, but it's down the hall from me, so I was quite lucky and paid attention to it. Torkel Klingberg, working in John Gabrielli's lab, just made this very odd discovery about that fractional anisotropy measure. He said, there's this one piece of the brain back over here in the posterior branch of the, he wasn't quite sure where it is over here, where if I look at the fractional anisotropy over here in this piece of the white matter over here, in the brains of good readers and poor readers, it turns out that the fractional anisotropy over here correlates with the reading skill of that person. And if you just think about that as a weird result, right? Because, here you are reading, you're reading some text, and then some guy puts you in an MR scanner and measures how the water flows around inside of your brain. And the way in which the water flows around in your brain over there, it ends up being predictive of how quickly you will be able to read. And that's a really kind of both grip. It's gotten a lot of attention, of course, and it's a gripping result. And what it calls out for is a little more explanation. So a number of us have kind of rushed in there. And I got into it. You can see Gabrielli's name is on this. And we started working on it ourselves. And I moved to doing this with children. Because, okay, maybe is this caused by years of poor reading? Or is this something that's intrinsic to the system that will limit you as a reader? So we moved this down to kids as young as 7 to 10. and so as young as 7 and over range up to 10. And we could replicate the result in kids. And we weren't the only ones. A colleague and friend of mine, Chris Bolio, unbeknownst to me, he became a friend afterwards, unbeknownst to me, had done the same study, because this Gabrielli result had really, surprised a lot of us. And this is from his paper, published independently of ours. And you look at these, it's like they're from the same measurement, right? There's good confirmation of the basic observation that Torkel and Gabrielli made. And we've replicated again, incidentally, for other reasons. So at least it's a result that we think is true. And we just have this is from a summary paper that Michal Ben-Shahar, that just came out, actually, in Current Opinions in Neurobiology, and showing several groups finding the same location in various studies in different groups and so forth. So there's something going on there. Okay, here, by the way, are the data showing fractional anisotropy and reading score. That's pretty good, and this is for the children. Less of a perfect correlation here, but they do separate out. But here it just hugs the line, and here it's more like there's 2 groups or something. So we're thinking about that. Okay, and that's only using FA. Right? I've got this whole technique here where I've got these tensors and shapes, and I've only done the first little goofy approximation of using FA, of just how distended is this thing. So what happens if we look at direction? Well, here's the original T1. Here's a region that was funky in the poor readers. Here's what it looks like in an FA map right over here. If you paint it with color, green indicates the places where the water is flowing superior, inferior. Blue, anterior, posterior. And red, right, left, like that. And that's the region there. And if you actually do the statistics on direction, not a significant difference back over here. And you can see it's a messy area. There's red, green, blue, all kinds of colors in there. But it turned out that there is a statistically significant direction difference over here in this part of the brain. Pretty far away, but a direction difference over here. So now that's kind of funny, right? You have an FA difference in one part of the brain, a direction difference in another part of the brain. And you start to think, well, maybe I shouldn't be thinking about these as little local things, but I should be thinking about these in terms of the natural structures of the brain, the actual fiber bundles. So we, and following many other groups, started, and Tony Sherbandi's here, and he's been working on this very intensely, has been trying to look at things like this. See, this one, this one, this one, this one, they're all pointing up and down. And say, well, okay, they're all telling me the same story. Maybe this is where I should say there's a fiber bundle like that. And then you can get a lot smarter at it than that and do it a few million times across the brain, and you end up getting estimates of where the major fiber bundles are, starting at different places and so forth. And I have a lot of good things to say about this particular fiber tracking, it's called, and some bad things. But some cases, if you know, we believe that there are some cases in which it can be very useful and other cases not so useful. And a case in which it's quite useful and has been very useful to us is if you find all of the fiber bundles that come through the corpus callosum. And that's a major structure of great interest. It tells you a lot about where the white matter is going in different parts. And you look at all of them, that's shown over here. And then you subdivide them by their projection zone, where they're headed to. Some are headed out here to parietal or occipital or temporal, and so forth, frontal. And then you sort of stain them. So these fibers here are now been stained by the color of where they're headed, their projection zone. You get a kind of functional grouping of these fibers. And and you can do this. This is done on 49 children, and you can segment out the callosum. And I'll be darned if Bob Doherty didn't find yet another dependence on reading. This is a measure of reading skill, in which one can look at the fibers in the corpus callosum headed to the temporal lobe and measure in real units, by the way. This is the thing that's got me most excited. See, this is no messing around here, none of this relative signal, blah, 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 stuff like that. This is units of squared microns per millisecond, how much water is diffusing transversely to the fibers in that part of the brain in these kids in real units. And you get a correlation between the diffusion of the brain of the water through these fibers and their reading score over here. It's like a third one. And this explains something like 28% of their reading score. When you think it's a little set of fibers like that. And it's the third one that we've got. And this will come out in PNAS in the next couple of weeks. So what you think you need then is a story that explains how all of these fiber tracts go together to give you all of these results. And normally I would have brought up, let me see if I actually have it. Okay. I don't have the perfect picture here, but I've got a little bit of a picture of these fiber tracts. And we think that what is going on in these brains is basically that in the poor readers, this fiber tract here, the red one, goes from right to left between them, grows exuberantly, more than it should have during development. As a result of that, this blue one over here is moved out of its natural position, maybe a little bit to the left and a little bit forward. That causes a change in the FA back over here. The exuberant growth over here of the red one is we explain by saying that's the measurement that we're seeing in the corpus callosum. The exuberant growth changes the radial diffusivity of these things. And the third bit about the direction, well, when this thing moves forward, it's a consistent tract, and it goes from being inconsistent to consistent as this moves forward, so we need to pick up the directional difference over there. And I don't know if that's true, okay, but I wanted to give you a sense, and we haven't published this, but I want to give you a sense of the way in which we're trying to reason about these things. We see it not as one thing, but there's a collection of these results. We think it has to do with the development of these pathways, and we think it's great that we can start measuring them and starting to see these pathways in this way. So in summary then, diffusion tensor imaging can be used in a variety of ways, just from the FA maps to all the way to fiber tracts. There's a lot of complexity to that technology. This isn't the kind of talk where I whine about all the bad things and good things. I'm happy to whine about that later, but I just didn't want to do it in full throttle in this talk. And interpreting these differences this actually goes to behavior and kind of substantial behaviors. We're very interested in the possibility of a lot of kids, not a huge number, but a fair number of kids. It's surprising how many just have a hard time ever learning how to read. It's just and there's nothing wrong with that kid, basically. Just the reading task is a really hard task. These are very bright kids, motivated, all kinds of great stuff about them. Some of them learn to read. They're just a little delayed. And some of them never learn to read. And if we could sort those out by these kinds of measurements, the one who, why put the family through 10 years of therapy and accusations and stress? If you know they're not going to learn to read, go get the tool to help you get the information another way. If we could help with that, that would be really cool. And so all the group here that I'm just showing you worked on all these parts of it. And this is Elisa, who worked, she's going to be at UC Irvine shortly, and she's worked on the maps. Bob Doherty's worked on everything. Oh, Tony, you're not up here. But anyway, Tony's right there. And Michal Ben-Shachar and Gail Deutsch worked on the reading studies. Nikos and Stelios worked intensely on the plasticity work with me. And thank you very much for your attention.
Question? Please.
Yeah, my name is Paul Bush. I wondered if in the lesion study, if you'd looked at any kind of changes outside of V1, because perhaps V1 is maybe more genetically or whatever, just more.
Yes, no, that's important. Let me just say that's important, and I do believe they're harder to either see or not see beyond V1. The receptive field sizes start to grow. But I actually, you don't even see the blind spot in V2. So in V1, you can see the blind spot pretty clearly. In V2, not. So changes are just harder to see. I've looked in humans, which is the direction I've taken. And I personally believe that the second, third, and cortical areas do reorganize more significantly to these missing spots. and we haven't published on that. The battle right now is about primary sensory areas. The other limit I want to say on our own results is, whereas I feel confident about this in V1 and have been urged on by others in this particular way, somatosensory cortex is a different story. I'm not talking about motor cortex. I'm not talking about somatosensory cortex. Everybody's got to look at their own piece of brain separately, and I'm not attempting to generalize. I really hope you'll go away and think I'm telling you about V1, and that's the end of it. And V2 and others, good question, other possibilities.
Two more questions.
Yeah, Cecilia Aragon from Lawrence Berkeley Lab. I have a question about the plasticity experiment as well. You applied the lesion to the retina of the macaque monkeys. Did you try lesioning that area of the brain but leaving the retinas alone? Because it seems to me that, when humans have a stroke, the damage is typically in the brain and not in the retina. Has there been work done kind of in the reverse direction there?
Well, stroke will be in the brain, but the number of disease cases of retinal disease versus central loss is the other way around. So, in other words, retinal dystrophies are enormous. In fact, I'm sorry to have to report that for those of us who are approaching 70, 1 out of 3 people beyond the age of 75 lose central vision for age-related macular degeneration. And nonetheless, as a scientific question, independent of who's got more or less, as a scientific question, there surely Stelios himself has done lesions of V1, and we have every reason to believe that following damage to V1, V1 itself, of course, does not reconstitute; it's gone. But that there is cortico-cortical. This is kind of like your question here. If you delete the V1 signal, what happens in the secondary and tertiary visual cortices? And it is my belief, but we are not ready to show you data on that there will be more reorganization in cortical circuitry than there will be in geniculocortical circuitry. So was that an answer to your question? You look a little like...
I didn't understand that last sentence.
When you do the lesion, so geniculocortical means from basically the retina to the LGN to V1, that seems pretty stuck. But if you mess up V1, it projects to V2. The part of V2 that waiting for its signal from V1, we believe will reorganize more. But I don't have the goods to show that.
Okay, thank you.
One last question.
This is more of a speculative question, but it's one I'm extremely interested in. The rate of autism in children has gone up by hundreds of % just in the last few years. This is just a wild guess on my part, but it seems to me that looking at these kinds of pathways, is there any thought that you have about maybe looking for a link between autism and similar kind of anomalies in the brain? Is that something that has crossed your mind or other people in your field?
Yeah. But we're going from, Brian speaks about stuff he knows about. Right. To
just a speculative question, that's all.
Yeah. It turns out an old high school chum of mine is a distinguished professor back at Rutgers, and at R.J. Johnson. I don't know, he's a big cheese, and he studies autism the whole time. And those guys are all over, and Eric Corshane down in San Diego, and those guys are all over the question of whether you can use these kinds of neuroimaging methods to understand autism. And in fact, the exuberant growth hypothesis for explaining autism I actually lifted this hypothesis from that field. So on the other hand, there's not that many people who are really, it's a funny thing. They've oddly started to ask me to review grants in that field, so maybe I'll learn something about it. It seems to me like a very important question.
Yeah, the reason is because there's been a rate of increase in autism. It's dramatic, and that was kind of my thought.
Yeah, and there is a hypothesis that there's a mismatch between the rate of growth of the brain and the rate of growth of the white matter fibers that are causing this. And it seems like an important question. I wish I knew more about it.
Thank you so much. Let's
Thank you.
One more time.